Hi everyone, I'm Abhilasha from IIT Kanpur and I'm doing research study over there. So basically I'm studying about the use of rice straw biochar as a material to prepare fertilizer. So in this particular presentation, I'll be discussing about the magnesium modified rice straw biochar based alginate beads that I'll be using as a phosphate fertilizer for Cicer arietinum plant, and that is called the Bengal gram in common language. So my research is inspired by two major problems that we are facing in agricultural field. So first one is the biomass burning. So what happens when the on every onset of winter, when the rice is harvested, there are stubbles of around 2 feet left out in the field, which are exposed to burning rather than collection and baling out, because it's a pretty expensive option for the farmers to do so. And as a result, what happens that there is a lot of generation of particulates, aerosols, and various oxides of like carbon dioxide, nitrogen oxides that pollute the environment, not only in that particular area but also in the other regions as well. So and not apart from polluting the environment, it is also damaging the soil structure, because the fertilizers that are present in inherently in the soil, like nitrogen fertilizers and all, they get released in form of nitrogen oxide, sulfur dioxides, and most importantly, the biota that is friendly for the soil, that is uh, dead because of the increasing temperature. So this is a screenshot from Indian newspaper that shows that 35% of share of the total pollution happening during the onset of winter is due to the farm fires. And this is the picture of a very popular signature bridge. You can understand the gravity of the problem, as the visibility is almost negligible in the picture. So this is the first issue. The other one is the agricultural runoff. This is another a major problem, because when the farmers add inorganic nutrients to the soil, they are exposed to, as soon as they are exposed to rainfall or excessive irrigation, the nutrients that are uh, uh, in the soil, they get dissolved and end up into the nearby water bodies. And it's like sorry we can see that it will end up into water bodies and lead to algal blooms. And other thing is that nutrient leaching is another problem that we are facing because of that. So um, for this particular study, why we have chosen the rice straw, as I told previously, that the rice straw is being during the onset of winter, whatever crop is there, that is leading to the maximum fraction of biomass burning. So rice straw is the major biomass during that time. And the other reason why it is being not bailed out by the farmers, because it fetches less commercial value in return of removing it and bailing it out. So the uses where we can possibly use the rice straw is, first one is the for cattle feed, and it is not a very suitable option because it is having low nutrient content, and only 2 to 7% of crude protein is present in the rice straw. When we compare it to other biomasses, it is comparatively low. Then high silica content, it leads to the formation of urinary calculi if we feed the cattle rice straw in excess. And it is because of this, the digestibility, because of presence of silica, the digestibility of rice straw is even low. And the fourth thing is that high oxalate content. So what oxalate leads to is increase in the pH of urine of cattle, and this increase in pH washes off the calcium from the body of the cattle, which is again not a very useful thing. So all in all, if cattle has to be fed by rice straw, it need to be supplemented with other nutrients, or something like we have to add some supplements to it. Otherwise, it is not a very suitable option. Then there are other industrial applications, like it is being used as a filler material in transporting goods, making rope, board, mat manufacturing and all. But before before making these products, it need to be processed. And in processing, when it is exposed to the spare part of the machineries, they get damaged very soon and need to be replaced frequently. Then there are certain agricultural applications, mushroom culture, thatching, like covering the rooftop with the rice straw, bedding material of animals, and composting. So these options don't fetch a lot of commercial value to the rice straw. And in case of composting, the degradability of rice straw is very low. So it is not performing very well in that situation also. So as we can see, there are a lot of limitations the rice straw is having, but still we can make use of it in other field. Like silica fertilization is nowadays very popular, because various literature studies state that if silica fertilization is applied along with the other nutrients, the uptake capacity of the plants increases, and the resistance to various disease is also increased. So the objective of my study is to prepare a rice straw based biochar fertilizer that is having fast nutrient uptake capacity, but at the same time, when it is applied to the soil, it releases the nutrient very gradually, slowly. So uh, like that's what we want. And the other uh, part of the study is the pot experiment, to see the feasibility of this material as a fertilizer. So I'll very briefly go through the initial part of the study, that is the biochar preparation. So first, the rice straw is collected from the field. It is sliced into 2 to 3 centimeter size and washed, so that the dust particles can be removed, the whatever mud is attached to it. Then it is soaked in magnesium solution. Then biochar is prepared at 600 degrees Celsius, the pyrolysis temperature, in the nitrogen environment. Then the alginate composite is prepared. The reason to prepare this is like the rice straw biochar that we attained is very flaky and fragile in nature. And if we use it directly, like in bulk scale, while handling, it will generate a lot of particulates. So it is not a very feasible thing to do. So we molded it into beads by using alginate as the binder. So here we studied the maximum uptake capacity of the beads. That is by studying the Langmuir isotherm. We found that the 64.39 milligram per gram is the maximum uptake capacity of these beads, which is significantly high. And the STEM analysis shows that the morphological structure of the beads, and the EDS analysis shows the major components that are present in the beads, that is carbon, oxygen, and after magnesium modification only, the magnesium also came. And on the right hand side, when phosphate is adsorbed onto the bead, we found the phosphorus signals as well. And here we can see that there are tiny crystalline kind of structures are appeared on the biochar if we compare it to the previous picture. So now comes the main application part, that is the slow release mechanism of the biochar. So that is our main objective of how the this material will be applied to the field and how it will be beneficial for the plants. So when these biochar beads are added to the soil, there will be some soil water around it when we apply water. Then the nutrients will be released, whatever nutrients, like here we are studying about the phosphate release, but whatever nutrients will be loading onto it will be released in the water around it till the equilibrium is reached. Simultaneously, these nutrients will be taken by the plants and root zone of the plant and will be supplied to the subsequent portion of the plants by the ion exchange process. And again, the gradient will be created around the beads. So this cycle will go on again and again and will release the nutrients in a very regulated manner. So to study this phenomena experimentally in the lab, what we did, we took these centrifuge tubes, added phosphate loaded beads to it, and synthetic groundwater is added to these tubes, and it is like it is given a equilibrium time of around 48 hours, so that the phosphate present in the beads is completely uh, like whatever portion is suitably released in the solution, and then after 48 hours the beads the synthetic groundwater is analyzed for phosphate. Again, after completion of that cycle, the synthetic groundwater is refilled and again waited for equilibrium to be attained. So this cycle will go on again and again till the complete phosphate stock is exhausted. So here we can see the results of that study. So each bar represent one cycle, and this phosphate concentration, initial concentration taken to load the beads was 20 ppm, and solid concentration was 4 gram per liter. So total phosphate uptake during this process was 0.966 milligram. Then the beads that are loaded with this much amount of phosphate are allowed to release the nutrients in the synthetic groundwater, as I showed previously. In each cycle we can see the amount of phosphate that is being released, and it is not a and we can see the release of phosphate is not in a very abrupt fashion. It is almost coming in a similar pattern. So 100%, nearly 100% of phosphate is released in water in 17 cycle with the duration of 34 days. And this is the cumulative amount of water that is added throughout these cycles, that is of 800 ml. So now to test the suitability of the fertilizer, we did pot experiment in which we uh, performed 3 treatments. First one is the control. Second one is the T3, that is of commercial fertilizer, and T4 is having the weeds that are loaded with phosphate. And growing medium chosen is coco peat because we wanted to choose a medium which is having less nutrient content, like especially the macronutrients are in negligible amount, while still it can provide us with some micronutrients for plants to grow. And plant chosen here is the Cicer arietinum. The reason is we wanted to test a phosphate fertilizer, and this plant being a leguminous plant, it fixes nitrogen of its own. Still we need a limited amount of nitrogen to be added. Then the duration of this experiment is of 52 days, because we wanted to extend it further, but due to sudden lockdown we had to pack the experiment within 52 days only. So like there are certain limited results are only being showed in further slides. So for this treatment, in every treatment, what we can see is there are 5 pots for each treatment, T1, T3, and T4, and each pot is having 4 saplings. So in total, per treatment, we have analyzed 20 plants to attain a statistically feasible data. Then these are the treatments, and what all things are added to the to the pots is being shown here. So for control only water is added. For commercial fertilizer, water, ammonium sulfate, single superphosphate. This is our major commercial fertilizer that we have added, and this is magnesium oxide. We are adding this externally because in our phosphorus loaded beads, the magnesium is already present. So the amount of magnesium that is present in phosphate loaded beads, we added similar amount of magnesium oxide for the T3 treatment. Then comes the T4 treatment, wherein we have added the beads along with ammonium sulfate and water. And the nutrients that are added are as per the recommended dose of fertilizer. So these are the picture of pot experiments. In T1, that is the control, we can see that the leaves are scantily populated and the height attained is height and density of biomass is not very high. While for the commercial fertilizer, the leaves, the number of leaves, the leaf density, and the biomass attained is comparatively higher than control. But we can see some pale leaves in some of the pots. And for the phosphate loaded MgO beads, we can see the biomass obtained is higher. The length of the color of the leaves is also appearing more healthy. So the result of pot experiment are on the basis of the height that was height and the number of branches that was measured on every 10 days of the uh, uh, every 10 days cycle. So when we see for the commercial fertilizer, the increase in height compared to control is 55.36%, while for phosphate loaded beads, it was 115% compared to the T1. Then the branches are declining. So basically, we can't say that number of branches is an absolute measure, because we were unable to measure the weight of the biomass of the dry biomass. We could not measure because of sudden closure of the experiment. And the number of flowers was measured only on the last day of the experiment, because the flowers appeared in last 5 days of the experiment. Then these are the picture of the plant roots. So here we can see these are the plant root of the control. In each pot we planted 4 plants, 4 saplings, and here we can see the root is like root for treatment, control treatment and commercial fertilizer treatment. The root were totally separable, like we could collect the 4 different roots for all the 4 plant in each pot. While for the T4 plant roots, we could not separate the roots because it was highly entangled, and we can see the health of the root is also really good. And here the picture of commercial fertilizer plant root is missing because like it's not with me right now, so I could not project it over here. While it was a bit healthier compared to T3, that is the only sorry, control, a bit healthier than the control, not very significantly high density. So I'd like to conclude my study with respect to the slow release fertilizer, that even the low initial phosphate loading, the release of phosphate is very slow, and it took 17 cycles to completely release the phosphate with the duration of 34 days. That shows that the amount of release of phosphate is so less that it cannot be like ending up into the water bodies, or it will not dissolve in such a high amount that it can be wasted. That means the nutrient use efficiency of the plant can also be increased. Then with respect to the pot experiment, I would say that the biomass gain and the root density is significantly high compared to the commercial and controlled treatment plants. And for this uh, and regarding the health of the biomass and root, the P-loaded magnesium oxide biochar is again leading the show, because even though we applied the nutrient as well as water in same amount, but the gain that we are getting in return is comparatively high. Thank you very much. Those were the experiments, and I'd like to thank my thesis supervisor, Dr. Purnendu Bose, and my colleagues who helped me with the experiments, especially the pot experiment, Madhukar and Manvendra, and seminar organizer, Christian, for organizing this wonderful talk. And the whole thing was not possible because of all the people about whom I have mentioned over here. Thank you very much.
Thank you very much, Abhilasha, for your nice presentation. And I'm really sorry to hear that your plant trial was suddenly stopped because of the lockdown.
Yeah.
So I would like to invite everyone to ask any questions. So you can either just unmute yourself and ask the question directly, or you can write in the chat. So I can ask then. And I actually have one question myself. So
yes.
How did you produce the beads from your biochar? I didn't get that point. So what did you do to produce the beads from the normal
the rice straw biochar was very flaky in nature, so we thought of binding it into a material. So these beads, the rice straw is actually crushed into fine powder, and then along with alginate, we make a like mixture of it, the rice straw biochar and alginate, and then we drop it into calcium chloride solution drop by drop, so that the spherification of those beads takes place. And further it is dried at 45 degrees Celsius.
Okay.
I have a question.
Yes.
Very interesting. What would you need to do to understand the economics to the farmer of using this fertilizer compared with what they're doing now? That is to say, not getting paid for larger environmental effects, but what would be the economics to the farmer? What would you need to do?
So actually what I'm thinking of doing is to get this get some decentralized units prepared in the farms, so that the biomass is collected then and there itself, and the biochar can be prepared out of it, and it can be used in the field directly. Like, we don't want to transport the material to long distances, and that will be another add-on cost, because the density of biomass is very less, and if we are carrying it to long distances, there are issues like the cost of transportation will be very high. So if we make decentralized units at the farms itself, so the farmers will have the opportunity to get employed there, other people in that area other than farmers who are interested in that thing, they can get employed in those biochar preparation plants, and at cheaper rates we can prepare the biochar at the site itself. Was I able to answer your question?
Thank you. I'm wondering how it translates to the farmer's rice crop. Where would the farmer use this, this legume in the planting cycle?
Pardon, I can't get it. Hello?
You're demonstrating a gain in the production of a legume using the biochar fertilizer from the rice stalk. Where does the legume fit into the farmer's process?
Actually, this is just to, because what I'm doing for this particular study, I've just made a phosphate fertilizer. So for that reason, I'm using it in using a legume to study that thing. Because for other plantation, maybe nitrogen is also very necessary. So in that case, sorry, the voice is getting reflected. So basically, the gram needs phosphate fertilization more, and it's not just related to the rice straw directly. The phosphate fertilization is the major factor that is responsible over here.
Thank you.
I would have another question for you, because we did some plant trials with biochar particles, and we observed that for certain biochars, the roots actually grew around the biochar particles. Did you observe the same thing when you took the plants out of the pots?
I'll show this picture for that. So probably here you can see that like I didn't observe that thing especially, but yes, we can see that this is the biochar bead, and it is growing well around it. I mean, without biochar, the roots are really scanty, as you can see over here. I'll zoom the picture. You can see here, this one. Like the roots are very thin for the control treatment and for the commercial fertilizer as well. But while for this, the one that is having biochar, these roots are really thick and very healthy. That's the difference I found.
Okay, thank you. Anyone else with a question for Abhilasha?
Yes.
Ah, yeah, okay. We actually got a couple of questions. So, why does the number of flowers are low in T4? What does it indicate? That's one question.
Okay, so again, this is the problem of lockdown. We could not extend the experiment for a longer duration so that we can see the actual flowering properly. Even though here, whatever we observed, that is within the duration of 4 to 5 days, whatever flowers came, it is based on that. So I'd say that that is not an absolute measure again, because like after flowering, we have this stage of this fruiting that is more important. So if probably we were able to attain that stage, we could have been more sure whether like the productivity of the plant is really being benefited or not. So that is the reason I'm not focusing more on the flower. But yes, the biomass gain that we can see visually is good, like higher.
And we got another question. Are you considering repeating the pot experiment after the lockdown?
Yeah, that will be painful, but I'll not do that again. But I'm having some more things to test. Like I have prepared one more fertilizer. These kind of beads, slight different composition. So probably I'll be doing experiment with that. I'll try to verify that point with those experiments. Like I'll take it to the fruiting stage and then only I'll conclude. But for now here, I'll just focus on the amount of biomass that is gained for the root as well as for the plants, and the nutrients that are inculcated in the plant uh, with the like in all the experiments.
And another question from Henry. Which of the experiments had the best absorption capacity based on the isotherm used? I think he's talking about the Langmuir isotherms you showed.
Experiment had the best absorption capacity. So actually, okay, you are asking about the literature probably. I guess like some other uh, materials that have performed well. Am I right?
I would assume he was talking about the one slide where you showed your isotherm from the first experiment, not from the plant trial, but from the phosphate.
This one?
Yes, I guess so.
Yeah, so actually this is isotherm, and yeah, I got an information from him that actually there are even higher uptake capacity materials also that I have not mentioned over here, but yes, even better adsorption capacity have been attained for different modifications and different materials.
So did you actually try different producing different biochars? Have you?
No, I have not tried. I have just focused on making using rice straw as the base material and then doing modifications onto it. So I tried doing the modification with calcium. That was not very successful. So I just went for this magnesium modification and proceeded with it. Then with the pot experiment and all till that stage. And further I'll be trying some more like with rice straw as the base material. I'll be trying to inculcate more nutrients, different uh, nutrients, so that we can have a better fertilizer, not just a phosphate one.
Thank you. And I would say let's wait another minute if you get another question, and otherwise we are well over the time already. So if no one has a question, I'm sure you can write an email to any of the presenters if you have follow-up questions.
Okay.
And otherwise, I would say